Right, in this next video, we're going to be talking about some of the techniques that we can use in order to increase contrast in microscopic samples. Of course, we have to do this because most biological samples that we might want to look at under the microscope are going to have very little in the way of contrast, very little in the way of color. Most cells are actually transparent. Consequently, if we look at a living specimen underneath the microscope, we won't see anything as the light passes directly through that specimen. We can add contrast to biological samples, such as this cheek cell that we see in this picture right here, by fiddling with some of the light path properties of the compound light microscope. As we saw in the last video, we can set up either dark field or phase contrast microscopy, which will allow us to see structure within cells, be able to see cells a little bit more clearly without doing anything extra to those. Within this video, we're going to talk about the property of staining, where we can add chemicals which will also increase contrast. The drawback of this approach, however, is that by treating cells with chemicals to increase contrast, we're typically going to be dealing with dead cells after this process. Consequently, if our experimental goal is to be able to recover living samples after looking at them under the microscope, this might not be the best choice. So the principle of adding chemicals, the process of staining, is going to utilize two basic classes of chemicals that we can add to a cell. And the chemical properties of those chemicals will dictate whether or not they bind to or are repelled from those cells. We can divide these chemicals up into two basic classes. So the types of chemicals that we can use are referred to as basic dyes, which, as we can see, has a net positive charge, or acidic dyes, which has a net negative charge. We have a listing of some of the different chemicals that have either basic or acidic characters associated with them. Most cells are going to end up having a net negative charge associated with them, and so if we bring a positively charged basic dye, what's going to happen is that positive charge on the dye is going to be attracted to the cells. Likewise, if we have a negatively charged acid dye, what's going to happen is that by and large the similar charges between the dye and the cell is going to result that the dye is going to be repelled away from the surface of our negatively charged cell. We can see this in action right down here in this micrograph, where we've come by and we've stained a preparation of cells using the acid dye nigrosin, which is black in color, and what we can see is that the background of this slide is darkly stained and the cells show up as unstained holes in that stained background. This process is referred to as negative staining, and in negative staining we end up with the background stained and cells unstained. And this is extremely rapid procedure, as we were able to see in lab last week. Quickly allow us to be able to assess the size, shape, and arrangement of an unknown organism simply by adding one single chemical and then immediately going to look at them under the microscope. Anytime we are using a single chemical, a single stain, either an acid dye or a basic dye, we refer to this process as simple staining. We're going by and using a single chemical that may be soluble in water or perhaps in alcohol, and we find is that these dyes will tend to bind to certain parts of the cell. This simple staining procedure can allow us to quickly assess, as we said, the size of an unknown microorganism, the shape, and the arrangement of the cells. Sometimes we need to add an extra chemical to those. This extra chemical is referred to as a mordant, and all a mordant is a second chemical that's added to a simple stain, which can help to make the color a little bit more intense, perhaps by making it so that that chemical binds to the cells or is repelled from the cells a little bit more easily. Or sometimes, as we'll see in one special case, the mordant is actually going to coat the cell and make it thicker, and therefore a little bit easier to see. The mordant itself is not going to be giving the cell a color. It's simply going to be making it easier to see under the microscope and make the stain more effective. So here's an example of a simple stain using the basic dye crystal violet. This actually looks very similar to the bacterial species that we saw with the nigrosin slide 2 slides back. Crystal violet is a very useful stain. It's a single chemical that we can add to an unknown microorganism specimen, and we can quickly see that all of the bacteria in here are all extremely similar in size. We can judge their shapes. They all appear to be round, and we can also judge the arrangement. We can describe the arrangement of this unknown microorganism stained with crystal violet as being arranged in clusters. The other characteristic arrangements that we might be able to see are chains of microorganisms where they are linked end to end, or perhaps isolated where we find the microorganisms spread apart and with no particular association to one another. So simple stains are going to be using just a single chemical in order to be able to essentially stain all microorganisms with the same type of color. Differential stains are slightly different, and what differential stains are going to do is that if we have one type of microorganism, a differential stain may impart one kind of color to it, whereas a different kind of microorganism may react very differently to that same staining procedure. So differential stains are going to be used to separate microorganisms into different groups. The most common of this is the gram stain that we'll be practicing over and over in microbiology laboratory. And the gram stain is used to differentiate bacteria into two broad classes of microorganisms, which are referred to as the gram-positive bacteria and the gram-negative bacteria. We learned a little bit about the cell wall structure of microorganisms during the Chapter 3 YouTube videos from Bauman, and what we learned in there is that the gram-positive bacteria have a very thick layer of peptidoglycan, and the gram-negative bacteria have a very thin layer of peptidoglycan plus an outer membrane. And it's actually this difference in the cell wall structure, the thick layer of peptidoglycan and the thin layer of peptidoglycan, that allows these different microorganisms to be able to react differently to the gram staining procedure. Let's look at the gram staining procedure in a little bit more detail once we see how different these microorganisms appear. This is a picture that's taken from a different textbook, not from Bauman, but we can clearly see in here that this smear of microorganisms have been stained with different kinds of chemicals, and we can easily see in here that there are large numbers of scattered rod-shaped cells, and if we were to measure those would be about 1 to 2, perhaps 3 micrometers in length, and we can see that these rod-like organisms are primarily pink in color. And we can also see that there are some very dark purple round-shaped cells, which, if we try to get the big picture from this, we can see that the round-shaped cells tend to be clustered or touching one another by and large throughout the smear. The pink microorganisms are going to be the gram-negative microorganisms, and the purple ones are going to be the gram-positive microorganisms. And so if we're working with an unknown that's a pure culture, we would hope to see either pink or purple cells in this. What this image shows us right here is very rapidly that there's 2 different microorganisms at least in this bacterial specimen. We know it's not just one single species of bacteria. So here's the gram staining procedure. We're actually going to practice this a lot during microbiology laboratory. The first step is to come in with a basic dye, and it's the basic dye crystal violet that we can see right here. And if we have gram-positive bacteria and gram-negative bacteria, after staining them with crystal violet, what we'll find is that they are both going to be purple after this step. It would be as if we just had a single microorganism and stained it with crystal violet as a simple stain. After 1 minute of staining with crystal violet, the excess dye is rinsed off with a little bit of water, and then we flood the slide with a second chemical called Graham's iodine. And Graham's iodine here is a mordant, which is, we recall that term mordant, what that means is that it helps the stain to bind to cells. And if we continue to view a gram-positive cell after staining with the mordant, what we'll find is that gram-positive bacteria are actually still purple. If we had a gram-negative microorganism and looked at it after this step as well, what we find is that that gram-negative bacteria is also purple. So we actually have not got a differential stain here at all. All microorganisms actually are going to appear the same after the second step here of flooding it with our mordant. Well, what we do at this point is we come back and we want to get rid of the extra dye that's on here, and this is where the differential step comes in. If we come back and remove the dye from our slide by using a solution of alcohol and acetone. This is referred to as Graham's alcohol. What happens is that the thin layer of peptidoglycan that we find in a gram-negative bacteria allows the dye to be removed from the cells when we use this Graham's alcohol solution. And so if we were to come back and have a gram-negative bacteria and look at it after staining with crystal violet and then with the iodine and then washing it with the Graham's alcohol, our gram-negative bacteria would actually appear colorless at this point. Our gram-positive bacteria with a thick layer of peptidoglycan would remain purple. The thick layer of peptidoglycan is going to help the gram-positive cells to retain the crystal violet color. Now, one place where a microbiology student can go wrong with this is if the solution that's used in step number two, the mordant, is forgotten. If we stain a gram-positive cell with a basic dye like crystal violet so that it's purple, and then omit the mordant, what's going to happen is actually when we come to step 3, the Graham's alcohol will remove the purple color even from a gram-positive cell. And so the inclusion of this mordant is actually super critical to get good results from the Graham staining procedure. Well, after step 3, we can see that gram-positive cells would be purple, gram-negative cells would be colorless if we're following the procedure correctly. We want to be able to see gram-negative bacteria, and so what we do is we add a second stain, which is known as a counter stain. And we want a color that's going to be different from our crystal violet that we have over here, and so the counter stain that's used typically in the Graham staining procedure is another basic dye called safranin, which is pink in color. And so our gram-negative cells, which are colorless after this step, now become pink. The pink color binds to the gram-positive cells as well, but the purple color is very overpowering, and so all you see, even though you've stained it with safranin as well, is the purple color. So our gram-positives at the end of this procedure would end up being purple in color. Our gram-negatives are going to be stained purple up here. They will still be purple with the mordant, but they become decolorized with the Graham's alcohol and then counterstained with the pink color down here. So if we do this well, we should end up with gram-positive cells that end up purple at the end of the process and gram-negative cells which end up pink. Places where microbiology students can go wrong, it's very good to be able to troubleshoot this, is if the mordant is used for not enough time, what's going to happen is that our gram-positives will tend to be decolorized when we come with the Graham's alcohol. The other problem is that can arise is when a student uses either too light of a touch with the Graham's alcohol or too heavy of a touch with the Graham's alcohol, and gram-positive and gram-negative cells can come out somewhat aberrantly, somewhat incorrectly due to the use of the technique. Now, it's also sometimes not the fault of the student, not the fault of the microbiology student when they come up with results that don't actually bear out to what they're supposed to be. The best results with the Graham staining procedure are going to be occurring with young cells. These will work the best and give the best Graham staining results. A culture which is old, which has been sitting around for a long, is going to typically give kind of aberrant results. And so if you have a microorganism that you know is supposed to be Graham positive but it's coming out Graham negative, one reason may be because the cells are very, very old. May not be the fault of the microbiology student at all. Alright, so the Graham staining is one of the important differential stains that we use, and what it's going to do is going to separate out microorganisms into two classes, the Gram positive and Gram negative bacteria. The acid-fast staining procedure is another differential stain, and it's going to broadly break out microorganisms into two classes of microorganisms: the acid-fast positive microorganisms and the acid-fast negative microorganisms. The principle of it is much the same as we saw with the gram staining procedure, where we come back with a primary stain, which in this case is called carbonyl fusion, a decolorizing step, which is going to remove the carbonyl fusion from the acid-fast negative microorganisms, and it's actually a solution of acid that's used, hydrochloric acid, that's used to decolorize these. And then a counter stain is a contrasting color, and what's typically used for the acid-fast procedure is a stain called methylene blue, so that we can see the non-acid fast bacteria. We can see two examples of some acid-fast stained slides here. Up here in the top, we have a mixture of microorganisms. Here you see these purple ones. These are going to be the ones that have been stained with the methylene blue, the counter stain. These are going to be the acid-fast negative bacteria. And here, these kind of hot pink colored ones that we see throughout here, these rod-like organisms, are going to be the ones that are stained with the primary stain, carbonyl fusion, but are not decolorized with the acid rinse. And these are going to be the acid-fast microorganisms. Here is another smear where we can see a lot of kind of gunky material throughout here that's very difficult to see any sort of detail, but in the center we can see these this cluster of hot pink carbonyl fusion stained cells. And these are going to be the acid-fast bacteria, and here in this background blue material is other material that's not acid-fast bacteria. This is a differential stain. When the microbiology student is going to hear about the acid-fast bacteria, they're really talking about one small class of bacteria. These are going to be gram-positive bacteria. But when we stain them with the gram stain, these hot pink cells, if these were stained with the gram staining procedure, what you would actually see is that they don't get stained positive or negative at all. They're gram-positive bacteria in that they have a thick layer of peptidoglycan, but they also have an added chemical called mycolic acid in their cell walls, and this addition of this mycolic acid makes it extremely difficult for the gram stain reagents to get inside these cells. These are organisms, these acid-fast organisms with the mycolic acid in their cell walls, are a broad class of bacteria, primarily the mycobacteria, which will also be known as the acid-fast bacteria, and the one that's of primary clinical relevance to microbiology students, to allied health students, is the organism Mycobacterium tuberculosis, which is of course the causative agent of TB. So if you're in clinicals and you hear about acid-fast positive bacteria, acid-fast staining, you're going to be associating that primarily in the region of the country where we live as the causative agent of TB. So one additional differential stain that we want to be able to understand the principle of, and one that we're going to practice in lab a couple of times, is the endospore stain, which is going to be differentiating organisms into endospore positive, endospore forming bacteria, and endospore non-forming bacteria. It's another set of stains of basic dyes that follows the same essential principle of primary stain, which in this case is a dye called malachite green, a decolorizing step, which is water in this case, and a secondary counterstain, which is going to be contrasting with the malachite green. And in the endospore stain, typically safranin is used, much like the last stain used in the gram staining procedure. Over here on the left, we see a field of microorganisms, and what we see in here is essentially just pink. They're round-shaped cells, and this is actually probably Streptococcus or Staphylococcus, which does not form endospores, and the only color that we see in here is the secondary stain due to safranin. This would be interpreted as being endospore negative. Over here on the right, we see some pink rod-shaped cells which have been stained with the secondary stain only safranin, but we also see in the interior of many of these cells these dark green round-shaped structures, which are the endospores. The endospore, of course, has an extremely thick coat on it, which doesn't allow the secondary stain to get in particularly easily, but the combination of malachite green and heat forces the malachite green into the endospores, giving it this green colored appearance. So the presence of these green oval structures is going to end up being positive for the endospore stain. The microbiology laboratory also uses a number of specialty stains which are going to be used not really as in a differential sense, but perhaps to be able to highlight one particular part of a microorganism. One that's commonly used is the flagella stain, which is actually just going to be a simple stain. But it's going to have a mordant added to it, and it's going to be designed to highlight the structure that we learned about in chapter 3, the flagella, which is essential for bacterial motility or bacterial movement. Now, most flagella are going to be on the order of around 20 to 25 nanometers in diameter, and if you recall from our initial section in chapter 4 talking about the light microscope, we recall that the light microscopes that we use in the laboratory have a limit of resolution of about 200 nanometers, and this number, this diameter of the flagella, is far smaller than the limit of resolution in the light microscope. What does this mean? Is that if a bacteria has flagella and we stain it with a simple stain like crystal violet, because this number here is smaller than the diameter of, or smaller than the limit of resolution in the light microscope, what this means is we won't be able to see flagella under the light microscope. The mordant in this case coats the outside surface of the bacterium and the outside surface of the flagella, making it larger than the limit of resolution of the light microscope, and consequently the combination of the simple stain plus the mordant allows us to be able to assess the presence of flagella on the surface of these bacteria, allowing us to see the arrangement of the flagella, which can help us to be able to identify what an unknown microorganism is. If we were to repeat this flagella stain with these same bacteria but forget the mordant, all we would see would be the bacterial cells on this slide. The capsule stain is another set of staining techniques that can be used to help us to visualize a structure that may be found on some bacteria but not on others, and that's the capsule. We recall capsules from chapter 3. Capsules are going to be important protective structures that are going to be distinct from the cell wall that help to protect a microorganism from phagocytosis, or to perhaps help it to be able to attach. And bacteria that possess a capsule are frequently going to be more pathogenic than bacteria that lack the capsule. The capsule stain is actually a very clever application of two different stains, two different chemicals. It's a combination of an acid dye, which we recall has a net negative charge, and a basic dye, which we recall has a net positive dye. And we also recall that bacteria are typically going to have a net negative charge, so the basic dye binds to the bacterium and the acid dye is going to be repelled away from the surface of the bacterium. So it's this mixture of a basic and an acid dye that stains the background. The acid dye stains the background, the basic dye stains the bacteria, and the capsule is this halo that surrounds the bacterium. If we have a bacterium that doesn't make a capsule, what's going to happen is that this background staining dye, the acid dye, is going to go all the way up to the edge of the bacterium, and this halo will be invisible. So the capsule in the capsule stain is the halo that surrounds bacteria, that actually doesn't take up the acid dye or the basic dye. So this table just kind of summarizes the different kinds of stains that we can use in the microbiology laboratory. We'll want to be able to know what their applications are for and how microorganisms might appear to use them. For the purposes of any exams that we have in Bio 230, the ones that we'll actually want to know the specific steps for and how to troubleshoot are actually going to be the gram stain. We want to be real comfortable with the procedure for the gram stain for any exam that we have in Bio 230. The other ones we will practice in the laboratory, and we'll perhaps be able to see and talk about troubleshooting those within the context of the lab.